Can CAR T cell therapy be used to cure Alzheimer's disease? Hi, my name is Ava Romano, and I am a grade 8 student from W.H. Morton Public School. For this year's science fair, I am happy to share my new perspective on how we might use CAR T cell therapy to cure Alzheimer's disease. In my spare time, I love learning about human anatomy and physiology. Most recently, I've been learning about the lymphatic and immune systems. In doing so, I came across a new clinical treatment called CAR T that has been used to treat different forms of cancer. More recently, scientists have started to see whether it might work on other diseases such as autoimmune and infectious diseases. What I didn't find was any research considering using this therapy on a disease which affects millions of people throughout the world, Alzheimer's disease. I will provide you with a brief overview of the immune system, explain the technology behind CAR T, and then share my thoughts on why I believe CAR T may provide a future cure to Alzheimer's disease.
The immune system is made up of two systems, the innate and the adaptive. The innate immune system does not require prior activation by a toxin or other foreign substance. It is comprised of physical barriers, skin, and mucous membranes that prevent pathogenic invasion and specialized cells such as natural killer cells and phagocytes that sense microorganisms and signal a defense response. The adaptive immune system is comprised of lymphocytes that get activated upon infection of antigen-presenting cells, followed by the production of specific antibodies against the antigen. My research focused on the adaptive immune system because it is the one that is used in CAR T therapy. The two basic types of lymphocytes of the adaptive immune system are B cells and T cells. When B cells encounter foreign material, antigens, they respond by changing into plasma cells, which produce antibodies that bind to the antigens on the surface of a pathogen cell, like a lock and key. This sets off a chain of events involving other components of the immune system to eventually destroy the intruder. The body has many different types of T cells that can react against virtually any antigen and perform different functions. CD8 plus or cytotoxic T cells travel to the site of infection to destroy its target. CD4 plus or helper T cells assist B cells to produce antibodies and assist cytotoxic T cells in their attack. Derived from the same lineage as CD4 plus cells, another type of cell is a regulatory T cell, which turns off other T cells once the infection is over. They are like a thermostat, keeping the immune system turned on just enough. The immune system creates an army of T cells to patrol our body for signs of infection and other diseases. The T cells inspect suspicious cells, and when they find an abnormal one, immediately lead an attack on it while limiting the damage to the healthy cells. T cells have claw-like structures called protein receptors on their surface that latch onto antigens, which are protein fragments found on the surface of cells. When a receptor latches onto an abnormal antigen, like one found only on a disease cell, the T cell turns on and releases toxic chemicals that damage the abnormal cell and recruit other immune helpers to the area. With cancer, infected cells can develop ways to fool the immune system. They can disguise themselves as healthy cells. They can sprout so many antigens on their surface that the T cells are confused and cannot mount an effective attack. Or they can find ways to turn off the immune response against them. Scientists have discovered a way to equip the body to fight against these deceptive or elusive cells through CAR T cell therapy. This is an infographic summarizing the process by which CAR T cells are manufactured. If we think of vaccines as teaching the body how to fight an infection for when it will eventually be tested, CAR T is like giving the body answers to the test ahead of time. The process of manufacturing CAR T begins with the collection of the patient's own T cells through leukapheresis. The T cells are then transported to a lab where a modified and inactive virus that can't cause any disease is used to introduce genetic information into the T cell. The T cells are reprogrammed so that they now produce special receptors called chimeric antigen receptors, or CARs, on their surface. The newly engineered cells called CAR T are grown in a lab through proliferation, and millions of them are then infused back into the patient. Now, the new receptors enable the T cells to latch on to a specific antigen on the patient's tumor cells and destroy them. CAR T has been effective in treating certain cancers, but is unfortunately quite expensive, approximately $500,000 per patient. I have identified 3 important criteria for selecting other potential disease candidates for CAR T. 1. Disease pathology. Do T cells play a role in the disease? 2. Are there other options available to treat the disease? 3. Are there cheaper alternatives to treatment? In considering these criteria, I identified Alzheimer's disease as a potential candidate for CAR T. Alzheimer's is a chronic neurodegenerative disease that causes shrinkage of the brain, which affects a person's ability to understand, think, remember, and communicate. It is one of the most devastating and prevalent diseases in the world, affecting nearly 750,000 Canadians and over 44 million people worldwide. Scientists are still trying to understand its disease pathology. However, recent research suggests that the T cell may play an important role. There is no effective treatment, and the cost to the Canadian healthcare system is over $10 billion each year. The cause of brain damage appears to involve two proteins found in the brain called beta amyloid and tau. Tau is a protein that normally transports nutrients to keep brain cells alive. In Alzheimer's, tau proteins collapse and twist, preventing nutrients from reaching the brain cells. Beta amyloid clumps into plaques, which build up between neurons and eventually block signals. As the level of amyloid reaches a tipping point, there is a rapid spread of tau throughout the brain. However, beta amyloid and tau may not be the only factors involved in Alzheimer's disease. While plaques and tangles form in the brain, we still do not know what causes them to form in the first place. Researchers have recently found that an adaptive immune response occurs in the blood and cerebrospinal fluid of individuals with Alzheimer's disease. Specifically, cytotoxic T cells are activated in Alzheimer's patients, and these cells exist both in the periphery and in the brain in beta amyloid plaques. From studying autoimmune diseases, I learned that when cytotoxic cells function improperly, they can produce autoantibodies, which are antibodies that attack the own body cells. This gave me the idea that perhaps these cytotoxic cells are the reason behind the plaques and tangles. If I'm right, then CAR-T could prove as a useful treatment or potential cure. CAR-T can activate T cells to fight against cancers by genetically engineering specific receptors. My theory is that if the role of cytotoxic T cells in Alzheimer's disease can be confirmed, then CAR-T cell therapy can be used as a curative treatment. Based on my research, I have identified two approaches to treating the elevated levels of cytotoxic T cells which may be causing the disease. 1. Create autoantibodies, chimeric autoantibody receptor T cells, to attack cells that are producing autoantibodies that are causing tissue damage and suppressing regulatory T cells. 2. Reprogram cytotoxic T cells into regulatory T cells to produce an overactive immune response.
As mentioned earlier, I love learning about the human body. It is amazing. It is adaptive, reactive, and has the ability to cure itself. Unfortunately, many diseases have become quite adaptive themselves and are able to evade our immune system and its natural defenses. With further research and therapies like CAR-T, I believe we can equip the body to better help itself against diseases which currently have no cure. Thank you for watching.